Geeks! Welcome to another video. So today, in the sixth part of our bioinformatics series, we are going to do the similarity searching for proteins. In the previous two videos, we continued this topic where firstly we studied about how to do alignment and how to calculate alignment scores by calculating the matches, gaps, and what are gap penalties. And then in the previous part, we discussed about the different algorithms, the dot matrix, global and local alignments. In this final video of this topic, we are going to cover the proteins. So now, without much further ado, let's dive right into this video. Now, if anybody asks you a question for alignment purposes, which alignment sequence should you use? Should you use a protein sequence or should you use a DNA sequence? What would be your answer? Sadly, I've already put the answer, but hopefully you all said that aligning proteins is more informative. Now, why using proteins for alignment is a better idea? The reasons are, firstly, many changes in DNA occur due to mutation, right? So residues change very easily due to mutation, but that doesn't mean that it changes the protein structure or the coded amino acid, right? So many changes in DNA do not actually change amino acids, and therefore, if you want to actually see what structural changes or what changes have taken place, protein is a better option. Also, many amino acids share very similar physiochemical properties. So these amino acids, although the code might change from the DNA, they may be substituted by a similar, protein, and this may not be taken into account over here. So therefore, for protein, you get a better alignment score and a better idea. Okay. Now, what do I mean by getting a better alignment score and amino acids having similar sequence similarity? So see, we know that we have different amino acids, 20 major, which are classified into groups called the hydrophobic amino acids, polar amino acids, acidic, basic, and aromatic, right? Now, say imagine you had amino acid glycine initially. After the mutation, the amino acid glycine got substituted with amino acid valine. Now, since the physiochemical properties of glycine is very similar to valine, there won't be a very drastic change in the protein. Whereas, imagine a scenario where an aromatic amino acid like tryptophan gets converted into a basic amino acid, lysine. Then in that case, there will definitely be some kind of change in the property of the protein. So whenever we have similar amino acids substituting each other, the result is less diverse as compared to when different amino acids get substituted. So for amino acids, unlike proteins, these things have to be quantitated. For DNA, just because we have ATGC, there is no question of having a similar sequence, right? Whereas in amino acids, that is a possibility. And therefore, that has to be taken into account while you are calculating the alignment score. So let us see how these things are taken into account. Before going on to that, let us understand the two related terms, sequence similarity and sequence identity. We have also done this with respect to DNA. Hopefully, when I say these terms, you remember it. If not, go to the previous video and check it out. So sequence similarity is nothing but the similar sequence, right? The sequence identity is the same exact sequence. Now, for a nucleotide, for a DNA, since it has just A, T, G, and C, the sequence similarity and sequence identity is exactly the same, right? But for proteins, they are not synonymous, they are different. For protein, identity refers to the matches, the percentage of matches of the same amino acid. Say you have sequence 1 and sequence 2, protein sequence of organism 1 and organism 2. If you have, say, in the organism 1, the sequence is KRDETQ, and organism 2, it is RRDDSN. Okay, now when you align for the percent identity, you will say this is identical, nothing else is identical. So out of 6, 2 are identical. So that your percent identity would be 1 by 3 into 100, right? But percent similarity will be something different. How? See, percent identity says the exact matches. So exact matches here are only 3. Percent similarity in the other cases, see here you have K and R, that is lysine and arginine. Since both are basic amino acids, these are similar amino acids. So therefore, the similarity here is one similar residue. Then E and D. So E and D both are acidic, so these are also similar. So now if you count the percent similarity value, it will be higher, right? So percent identity and percent similarity are different for nucleotide sequences. Whereas for same for nucleotide sequences, whereas for proteins, they are different because we have amino acids that have different physiochemical properties, some of which are very similar to each other. Okay, now how to calculate the similarity and identity? You can use these formulae to calculate all of this. Now amino acid scoring and matches. So now let's look at the amino acid scoring matrices. So when we use the Needleman, Wunsch or Smith, Waterman, etc., there was nowhere where they accounted for this percent similarity because it was nucleotide. But now when you're designing matrices, okay, for scoring amino acids, you have to reflect the likelihood of residue substitutions. That is, when a similar amino acid has been substituted, you will still give it a lower value. Whereas if distinct are, present, then you will give it a more negative value. You will understand what I'm saying a little better when I show you this matrix. So just for now, let me show you one matrix. So see, here we have the aromatic amino acids, phenylalanine, tyrosine, and tryptophan. Phenylalanine, tyrosine, tryptophan. Then large hydrophobic amino acids are grouped over here. Basic amino acids are grouped over here. So each amino acid is grouped like this, and these amino acids are arranged on the two axes, that is the x-axis and the y-axis. Now see, let's look at a case where you have C substituted with C. Now, since both the amino acids are same, you have to give it a positive value, isn't it? Because both, it's an exact match in both the protein sequence, so it gets a positive value. To make it more easier, let me write this. Protein sequence 1, protein sequence 2. So let's say you have protein T, P, A, G, N, and you have other protein T, N, D, let's say V, and again P. Okay, now let's score this. T with T is an exact match. So T with another T, where is that? Over here. So you assign a value of 5, correct? Then you have P with N. So let's see for P with N. P with N. So it's -2. Understanding? -2. Now, why is the sign given -2? Because see, P is an amino acid which is what? Yes, it's very different from the amino acids N. So both of them have very distinct properties. So if they are substituted, then definitely that has to be given a negative score. Then A with the D. So where is the D? D is here and A is over here. So -2. Again, they have different properties. So you have minus value. G with V. V is over here, whereas G here is G. So see, the value again is -3. So anywhere you have a similar amino acid, you have a less value. For example, you had M and F. If you had M over here and F over here, they're not exact matches. But see, the value is 0. 0 means, it's still better. So some values are minus, some values are 0, some values are plus. So when do we give a positive score? When the frequency of substitution is greater than that would have occurred by random chance. And usually those that are similar are given this positive value. Then 0 score. 0 score is given when there is zero probability that this has happened by chance. And negative score is where frequency is less than that would have happened by a random chance. So the idea is that those residues that are similar, those amino acids that are similar, have a higher probability of substituting each other, right? So if we have the phenylalanine aromatic amino acid, the probability that it will get substituted by another aromatic amino acid is higher, right? Whereas the probability that phenylalanine will get, substituted with, say, where's phenylalanine over here, F, yeah, phenylalanine will get substituted with R, that is arginine, is like by chance -3 value that way. Okay, so these minus values plus values are the probability of these substitutions happening by chance. So if you have a positive value, the probability that it happens by chance is higher, whereas if it's a negative, the chance that it happens by probability is very low. So that is taken into account. So based on this, you have different scoring matrices. So those values that are assigned, how did they come to this value? That is, actually what the scoring matrices are. So scoring matrices are devised to reflect the likelihood of residue substitutions. Okay. Two types of matrices are present right now. One matrice is based on the interchangeability of the genetic code or the amino acid properties, and the other one is an empirical study. You can just say that one is like a theory based method and the other one is an actual practical observation. Means what? So theoretically is this method where you actually make a scoring matrix based on the interchangeability of the genetic code, meaning so AAG getting substituted to AA, say A. What is the probability of G interchanging with A? That is actually taken into account. Whereas in the empirical studies, they have observed different proteins. Okay, they have observed different proteins which are related and actually, counted the amino acid substitutions and then made a matrix. So quite obviously, this one, the empirical matrices are more accurate because they are based on practical examples like existing proteins and their amino acid sequence. So two examples of empirical studies include the PAM matrix and the BLOSUM. So now what is the PAM matrix? A lot of questions on this have been asked in the DBT. So please try to understand what these are. So under the empirical scoring matrix, we have PAM and BLOSUM. PAM stands for accepted point mutations. Okay, so PAM is accepted point mutations. You will say, but this stands for APM. Actually, they have just swapped the P for A to make it better sounding. Like PAM sounds better than APM, right? It's easier to say. So PAM stands for accepted point mutations, and this matrix was designed by Margaret Dayhoff. Okay, in this particular matrix, what we do? So it is derived, these values are derived by observing evolutionary changes among 71 closely related protein sequences. So what Margaret Dayhoff did is she looked at 71 related proteins and seen the evolutionary change. That is, she seen the substitutions in the amino acids and extrapolated these data into probabilities that you saw in the matrix just given below. Okay. And these probabilities were then correlated to the evolutionary distance. The evolutionary distance is given in terms of a PAM unit. So how can you define a PAM unit? A PAM unit is defined as 1% amino acid change or 1 mutation per 100 residues. So what is PAM? A PAM unit, that is a accepted point mutation unit, is defined as 1% amino change or 1 mutation in 100 residues. A higher PAM number correlates with increasing PAM units. So more the PAM number, that is 10%, 20%, more the number means more the increasing PAM units, and therefore greater is the evolutionary distance. Smaller numbers means they have shorter evolutionary distance, means they are more closely related. I can show you an image to make this idea clearer in your mind. So look at this table. You have PAM1, which is 1% mutation rate. That is, the sequence is 99% identical. Whereas as the PAM number increases, say it's gone to 250. So a higher PAM number means that there is higher mutation rate. Higher mutation rate means what? Many of the nucleotides, many of the amino acids have changed. and therefore quite obviously the sequence identity will decrease. So how do you read this? How do you read if they give you PAM 250? So PAM 250 actually refers to evolutionary distance of 250 accepted point mutations per 100 amino acids. Okay. So PAM, if I say PAM 80, so what would PAM 80 be? PAM 80 refers to evolutionary distance of 80 accepted point mutations per 100 amino acids. A lower serial number of the PAM means that it is more identical, that is, it is more closely related. So if you want to compare two closely related sequence, you should use a PAM matrix of a lower serial number. Whereas if the proteins are divergent proteins, say the protein of human and bacteria, and in that case you use a higher PAM number matrix. Okay. Then the next matrix available is the BLOSUM one. To understand the BLOSUM, let us understand some assumptions of the PAM matrix. I'll just focus on the important assumption. So first important assumption is that these PAM matrices, except for the PAM 1, all others are based on predictions. Okay, that is, they have studied the different proteins for the PAM 1. So they studied those 70 different proteins and made this PAM 1. After that, they have made PAM 80, 250, etc. based on predictions. Okay. When PAM 1 was formed, it was in 1970. It was for the proteins available up to 1978. So it was a very small set of proteins, and usually those proteins at that time were mostly globular. So the data that is observed for creating this matrix is limited. It is not a wholesome data, and therefore these two assumptions make the PAM matrix less accurate because of a smaller data set and because of these predictions. So therefore, to overcome this came the BLOSUM matrix. BLOSUM stands for Blocks Amino Acid Substitution Matrix. How is this matrix formed? It is a result of direct observations of every possible amino acid substitution in multiple sequence alignment. So they have aligned multiple sequence of proteins, and by directly observing it, they have come up with this matrix. So they have looked at more than 2000 conserved amino acid patterns. These patterns, these amino acid patterns, are what are called as blocks. They looked at these conserved sequences and they used this data to actually come up with the BLOSUM matrix. Okay, the frequencies of amino acid substitutions of the residues in these blocks are calculated to produce a number table, like how we seen in PAM, which is called as the block substitution matrix. So they have used the multiple sequence alignment of a greater number of amino acids. They have looked at amino acid sequence pattern called as blocks, and they have created a matrix to score. Okay. Now, BLOSUM matrix, unlike PAM matrices, are easier to read. Like, what does this number indicate? So BLOSUM matrices are actual percentage identity values of sequence selected for the construction of matrices. That is, if you are using BLOSUM 62, what does that mean? That means that the sequences selected for constructing the matrix share an average identity of 62%. So while they were making this, they selected protein 1 and protein 2 that had 60% similarity, and then they seen the relatedness. Similarly, if you have BLOSUM 20, so BLOSUM 20 means these two sequences were only 20% identical. So when you use a lower BLOSUM number, means the sequence is more divergent, common sense. So if you use the sequence from human and mice, you will use a BLOSUM of lower value, right? BLOSUM say 10, because these two sequences are very, very divergent. However, if you are checking between human 1 and a close relative, say a gorilla, then you may use a BLOSUM of say BLOSUM 60 or something, or higher value, because these sequences are more similar, okay, having greater identity. So if you want to check divergent proteins, use a BLOSUM matrix of lower number, whereas for similar sequences, you use a higher number. Now, quickly, let's summarize this. by understanding the differences between PAM and BLOSUM and when you should use what. So as we see how PAM matrix was derived by an evolutionary model, right? They looked at the different protein sequences and their evolution, and they created the matrix. Except for PAM1, the others are all derived. Whereas BLOSUM is a result of direct observations. Now, because PAM was derived using, similar protein sequences that have evolved from each other, they are very useful for construction of phylogenetic trees. So when you want to construct phylogenetic trees, then you should go for the PAM matrix. BLOSUM, since it has come from direct observations, you don't get enough evolutionary data. Okay, PAM is less realistic for divergent sequences. If you have sequences that are very, very different from each other, they obviously will not give you a good value, because PAM was designed on a data set that was an evolutionary model. So if you have divergent sequence, it's better to go for BLOSUM because it uses conserved amino acid protein domains. Okay. Then BLOSUM is more accurate and reliable because it is taken from a larger number of proteins, that is around 20,000, whereas the PAM was only on 71 amino acids. And also PAM was created from a data set of proteins during the 1978, which was a smaller and less representative data set. So BLOSUM is highly preferred in this case. But however, the end line is, if you want to carry out any evolutionary study, go for PAM. If you want to study out any divergent study or for any conserved motif, you go for the BLOSUM matrix. And these are some commonly used matrices. Like for similar alignments, PAM 40 is used, BLOSUM 90 is used for highly similar, for BLOSUM 62 for potential similarities, 250 for longer alignments of divergent sequences, and so on. So hopefully you have understood. All you need to remember is what these numbers indicate. Most of the questions are based on these itself. So that's it for me for this video. I'll see you in the next one. Bye.